This is our sixth and final of this season. So there's a preview that we are thinking. It's been so popular that we might be renewed by the network for another season of webinar series. So it's been a good event. So maybe we'll start and others can join, because I'm going to talk a little bit. I'm going to introduce myself. I'm Steve Webb, and I'm the CEO of the Global Institute for Food Security. And again, welcome to today's edition of GIFS Presents, our engineering biology speaker series. Background on GIFS: we're a public-private partnership that was established in 2012 between Nutrien, the province of Saskatchewan, and the University of Saskatchewan. And our mission is to discover, develop, and deliver innovative solutions for the production of globally sustainable food by working with partners all the way up and down the innovation value chain: industry, academics, government. We serve as agriculture and ag and food tech's innovation catalyst, connecting the ecosystem, advancing innovation, and helping bridge the gap to commercialization to deliver resilient and sustainable food security for all stakeholders here in Canada and around the world. As I've mentioned before, but I think it's really, really important, and I want to thank Dr. Angela Bernard-Hone, the Dean of USask College of Ag and Bioresources. Angela is the inspiration for this seminar series, and again, I want to thank her for putting the idea out there, and it's been great series of talks. This was created in large part due to the announcement that we made earlier this year about the federal government establishing the awarding us the CFI funding to build our engineering biology center focused on agriculture and food. And instead of having me talk about it, as I've mentioned before, it was decided that it would be much better to have people with real experience and expertise from across Canada and the US to speak in depth on the technology and on the scope it can have across various fields. Our last edition in this series featured Dr. Vincent Martin from Concordia University, and Vincent's also a member of the National Engineering Biology Steering Team here in Canada, and I have the pleasure of being a member of that team with Vincent. He provided a great overview of the application of the platform in his presentation entitled Drugs from Bugs. And he used a couple of examples to highlight applications of the platform to accelerate the design, build, test, learn cycle that we were first introduced way back from the very first speaker in this series, Dr. Patrick Westphal. He identified, I thought what was really important, some of the challenge of having a technical achievement, e.g. a peak on a mass spec or a gas chromatograph or an HPLC to delivering impact at grams per liter, that need to drive productivity to make it economically viable. He identified some of the challenges in scale up. He used some examples of on-pathway and off-pathway modifications that led to yield improvement, and also the need to develop additional infrastructure here in Canada to support the transition from benchtop fermentation vessels to pilot scale to be able to do optimization. And his video presentation, Vincent's presentation, along with all of the others, are available on the event page and on GIF's YouTube channel. And today, we're privileged to have Dr. Joerg Bohlmann, professor at the Michael Smith Labs at the University of British Columbia. Joerg is also a member of GIF's International Science Advisory Panel. That panel advises, conducts reviews, and provides feedback to our chair, the board chair of the Global Institute for Food Security, on the science, our science strategy, and the development of our people at the Global Institute for Food Security. Joerg's here to speak to us today about plant bioproducts, food and human health, with a specific example on secondary metabolite that offers promise as a potential new therapy for metabolic diseases, specifically type 2 diabetes and obesity. And Joerg, with that rather long introduction, I am going to hand it over to you, the expert, to present, and I'm very much looking forward to your presentation today. Joerg?
Great. Thank you, Steve. Thank you so much, Steve, for the invitation to present to this group today. And thank you for everybody at GIFS for a fantastic working relationship over the last several years. And many thanks to the previous speakers. I think with one exception, I was able to attend all of the previous talks, and I've learned a lot, and I've seen a lot of opportunities where hopefully some of what we are doing can plug in and can connect as a direct connection with the first presentation by Dr. Westphal and then last week's presentation by Vincent Martin. I think there's a lot of connectivity between what I will be talking today with a very specific example and the presentation that we have previously heard. So I talk today about a very specific example of a so-called plant specialized metabolite called MONT-A that can be developed into a potentially novel treatment to deal with type 2 diabetes and obesity. And that's in the more broader context, as Steve already said, of plant bioproducts in general. We heard beautiful examples last week from Vincent Martin about important plant bioproducts. But also falls into a context of food and human health. So I hope to just at least briefly touch on a few different aspects here, but I will keep a focus on this particular one molecule. So I go a bit back between general aspects and very specialized aspects with my presentation. I want to actually relate to previous week's presentation by Vincent Martin. Vincent spoke about bioengineering of high-value metabolites and gave examples specifically on plant metabolites. And he gave us the full pathway from metabolite discovery, metabolic pathway discovery, all the way to how their foundry can actually use genes that have been discovered for particular biosynthetic pathways to develop and improve yeast-based production systems. Vince touched a little bit on metabolic pathway discovery, and this is where actually much of our research is in an academic environment. Metabolic pathway discovery is by no means trivial. It's become much, much faster with various omics platforms over the last decade, but it's still not entirely trivial. I also want to acknowledge Steve with a few comments that he made last week and that he picked up as questions from the panel discussion. Steve mentioned that he appreciates the chemistry and chemical structures. And in the discussion last week, we heard about chemical diversity. And I would say actually plant biodiversity equals chemical biodiversity. And I think there was a question in discussion last week about biodiversity approach to genome sequencing to be able to actually capture this enormous potential of plant biodiversity that equals chemical diversity, that equals bioproduct opportunities. And what's known today is that there's approximately 250,000 metabolites that plants produce that have no obvious function in growth and development of plants, but that we would have traditionally called secondary metabolites, implying that there might be of secondary value. But we have now appreciated that these are extremely valuable metabolites, and we would no longer call them secondary metabolites, but we like to call them specialized metabolites. And Vincent gave us some beautiful examples last week with the anti-malaria drug. Artemisinin, he touched on Taxol, he talked about opioid type compounds, and there are like 250,000 of these compounds that we can predict. But in the unexplored biodiversity of the plant kingdom and the oceans with all the sea organisms, there's probably a much, much larger group of metabolites that have great potential to benefit humans. So this is my acknowledgement. I talk a bit of research, so I need to actually mention that much of the research that I'm presenting today and the results is work that was done or led by Dr. Sandra Irmisch. Sandra was a postdoc in my group for the last 4 or 5 years. She came from the Max Planck Institute for Chemical Ecology in Vienna, Germany, trained there with Dr. Jonathan Gershenden, then joined my group. And just as the beginning of October this year, she is starting her own lab as a professor at the University of Leiden in the Netherlands. Another big acknowledgement, Dr. Steve Withers. Steve is a chemist at the UBC Chemistry Department, a colleague who actually inspired this work that I will be talking about. And he inspired this work about for us to get involved about 5 or 6 years ago. Talking about diabetes as the background for our discovery of functions of plant metabolites today. Diabetes is the inability of the human body to control blood glucose levels. This results over periods of years to substantial damage to various organs of the human body. We distinguish between type 1 diabetes and type 2 diabetes. Type 1 diabetes is the inability to produce insulin, which regulates blood glucose level. And the much more prevalent form of diabetes in our societies around the world is type 2 diabetes, which is the inefficient use of insulin to control blood glucose levels. Diabetes is not a small issue. It's actually a disease that's affecting nearly 500 million people around the world. The trends are going upwards. And the majority of these cases are type 2 diabetes. In Canada, and this is the case for most developed countries, approximately 10% of the population suffers from diabetes. And again, majority is type 2 diabetes. So this is about 4 million people in Canada. Globally, it's among the top 10 leading causes for premature death, and it's truly an epidemic. It's often associated with obesity, and it's also increasingly affecting children and young adults. It is, this is interesting and important in the context of GIST, it's the central issue of global food security. Taking a look at the latest UN report on food security, diabetes and obesity are themes that run through the entire report. Obesity and diabetes are not just issues of high and middle income countries. There are issues of low income, middle income, and high income countries. And the different levels of food security or respectively food insecurity affect increasing rates of diabetes. It's, at least in our part of the world, in the high income countries, lifestyle epidemic, at least in part, often associated with overconsumption of sugar or carbohydrate-rich diets, sugary beverages, lack of physical activity. And therefore, the easiest way to deal with diabetes would be change lifestyle. But there's one thing that is extremely difficult to control in people, that's their behavior or to affect. So we need treatment options that go beyond healthy diets and physical activity, which would be the ideal way to at least prevent or reduce the incidence of diabetes. But one can actually treat diabetes with drugs and with nutraceuticals. And traditionally, the way to deal with type 2 diabetes has been to affect starch degradation or generally carbohydrate digestion. And this is targeting enzymes in our intestinal carbohydrate digestive systems, which starts with the breakdown of starch. Starch is being broken down by the pancreatic alpha amylase or human pancreatic alpha amylase into oligosaccharides. These oligosaccharides then are substrates for alpha glucosidases. The alpha glucosidases release glucose, and glucose can be taken up into the bloodstream. And the result is that after a carbohydrate-rich meal, you had just a pasta meal or your pizza dinner, you get a rush of glucose in your bloodstream. This is where insulin comes in to control this, but if your insulin system is not properly working anymore, you get an extreme sugar rush that's associated with diabetes if you're diabetic. Drugs that are currently on the market, such as acarbose or miglitol, inhibit preferentially the alpha glucosidases, which leads to a buildup of oligosaccharides. Now, these oligosaccharides then pass through the digestive system, and they become substrate for microbial fermentation. And that leads to very unpleasant side effects, such as bloating, flatulence, pain, discomfort, and that in turn often leads to poor compliance. So patients who are on these drugs may often decide that they don't feel comfortable taking them, that the effects are uncomfortable, that they're unpleasant, that they're painful, and then just go off the drugs and don't take them regularly. So an idea that was pursued by Steve Withers and his colleagues several years ago was to screen for inhibitors in the upstream breakdown of starch, specifically targeting the human pancreatic alpha amylase, which, if this enzyme is being inhibited or stopped in its activity, starch would accumulate, and starch is not a substrate for most of the gut microbiome and can therefore not be used for fermentation and would therefore not result in the unpleasant side effects that are associated with inhibitors of alpha glucosidases. Steve Withers and his colleagues undertook a large-scale screen. They actually screened over 30,000 accession of so-called natural products, which one can say is secondary metabolites or special metabolites. They had access to 30,000 chemical accessions that came either from established libraries, from their own collections, commercial libraries, private libraries or libraries that they built in their own laboratories. So they screened about 30,000 natural product accessions for inhibitors of the human pancreatic alpha amylase. And in this high throughput automated screen, they found one really, really, really good hit. And this really good hit was a molecule that had already been known, monopretin A. And this molecule has an extremely high efficiency in inhibiting the human pancreatic alpha amylase, the entry enzyme in starch degradation. For the biochemist in this audience, this is the Ki value, 8 nanomolar. That's an extremely low concentration at which this molecule is active at inhibiting the enzyme, so extremely efficient in inhibiting this enzyme. The molecule is very complex. We don't need to be chemists to actually see that there's multiple parts and components in this molecules. We often look at this as a Lego design where we have our Lego building blocks and they can be put together in various forms. So we have a core structure here that's called myristoyl. This myristoyl is decorated with various sugars in its side chains, so 2 side chains, one with 2 sugar xylose and rhamnose, and another side chain composed of 3 sugar molecules, 2 glucoses and 1 rhamnose. And then here we have a caffeic acid side chain that is derived from phenylpropanol pathway. So again, you don't need to be chemist or biochemist to recognize the complex molecule. This molecule was known before, but its biological activity as an inhibitor of the pancreatic alpha amylase had not been known. This was a discovery that came out of this particular study. This molecule, as the name says, comes from a plant called Montbrechia. And summer is for most of us in the past, unless you're now in your southern hemisphere. This is the plant that you may remember from your summer days. It grows actually very abundantly, at least on the West Coast gardens here, but I think you find it very much around the world as a horticultural plant called Montbrechia or Cocosmia. This is actually the only known source for this particular molecule. It's native to southern and eastern Africa. And here's part of the challenge already. But I'll tell you about the opportunity first. So this molecule was discovered as having this fantastic activity in inhibiting human pancreatic alpha amylase. And Withers and his colleagues saw it as an opportunity to develop potentially a new pharmaceutical or a nutraceutical based on this molecule. They launched animal studies that have been completed in wild type and diabetic rats. They completed the animal toxicity studies. All of these studies were successful. And based on the animal studies in rats, Health Canada approved this molecule now for phase 1 clinical trials. Those clinical trials should have been completed already, but we were delayed by COVID. There was just no capacity to test this molecule at the time. But hopefully we can go into the clinical trials now very soon. So here's the opportunity, here's the problem. It's an extremely rare molecule. I mentioned already it's only this particular plant, Montbrechia, that is known to produce this compound. Within this plant, it's only been found in underground storage organs that we may call bulbs today. A specific technical name is corps. They look a little bit like tulip bulbs, and you can buy them at this time of the year in your local garden center. You can put them in the ground, and next summer you will have this plant growing. It's only in these below ground starch-rich storage organ where the plant produces, this particular one plant species produces this molecule in very low amounts. This molecule is too complex to produce this through chemical synthesis, and hence there is a need for alternative production systems. This is where perhaps bioengineering comes in. So, but this first is where Steve Withers came in again, where he engaged me in a discussion. And I remember the day when he knocked at my door and we talked in the hallway. Steve is a fantastic chemist, synthetic organic chemist and biochemist, but he knew this molecule cannot be efficiently produced by synthetic organic chemistry. So he was curious, how does then the plant produce it if chemists cannot produce it? And if we find out how the plant produces this molecule, i.e. if we can figure out what the enzymes are that are involved in the biosynthesis, and then by extension what the genes are that encode for these enzymes, could be used, the tools from the plants, the genes and the enzymes, and can be used for a metabolic bioengineering approach. And yes, this can be done. And Vincent Martin and several others and myself have been involved in these type of approaches over many years now, discovering of metabolic pathways and then utilizing these discoveries of genes and enzymes towards bioengineering. So I laid out in the conversation with Steve what our very general approach would be. And at that time, nothing was known about the biosynthesis of the molecule, but the molecule was known. So to go towards metabolic engineering as a base for bioengineering, we need the genes and the enzymes that produce this molecule. So how do we get to the genes and the enzymes? We took an omics approach where we combined metabolite profiling and transcriptome profiling, correlated data on the spatial and temporal patterns as to when and where this particular molecule occurs in the plant, with features of the transcriptome that have matching temporal and spatial patterns. Use this, those data for bioinformatics approach, for candidate gene mining, then functionally characterize the genes, and now we have them in hand for metabolic engineering and going towards bioengineering. So that's a very highly simplified discovery strategy that was definitely sufficient for the hallway discussions that Steve and I had, but it was simplified. And following this was a few years of work where we went into the details. But the short of this is, so this is now 5 or 6 years of work with several students and postdocs in our group. We went from looking at metabolite features, looking at the chemical structures, predicting theoretical biosynthetic pathways, measuring, detecting enzyme activities in the plants that could encode for or that are involved in the biosynthesis, going through the gene discovery strategy, as I had laid out, that is a combination of transcriptome profiling and metabolite profiling, sequencing candidate genes, selecting candidate genes, cloning them or synthesizing them, expressing these candidate genes in various heterologous systems. I'm only mentioning E. coli here. We used also E. coli, yeast, and a laboratory, a form of tobacco, Nicotiana benthamiana, that is widely used now for pathway discovery and potentially also for production of metabolites. And eventually we were able to reconstruct the metabolic pathway for this compound montbretin, both in yeast and in Nicotiana benthamiana. One slide, several years of work discovering a metabolic pathway that was not previously known. What made this actually quite challenging was that at the time, none of the metabolite standards that were potential intermediates in the biosyntheses were known. So none of the intermediates were known or commercially available, and thereby by extension, obviously there was no commercial standards available for any of our chemical analyses. All of these had to be made in collaboration with the chemistry lab, basically by taking the intact molecule and breaking it down stepwise. No prior biochemistry or gene discovery had been done in this plant, and that's actually very important for this general approach of exploring biodiversity for bioproducts. We are not working with model systems in the majority of these cases. We are typically not working with systems where genomic resources are already available. So taking the hint from last week's discussion, a biodiversity-wide sequencing approach is really giving us resources for gene discovery towards bioengineering. So these are not model systems in which we discover new biosynthetic capacities. And it wasn't just one more gene to discover, but it was actually discovering and reconstructing a biochemical pathway system. Short is, after 4 or 5 years of work, and this is where Sandra Irmisch was really leading the research in the lab, she discovered all of the genes and the enzymes that are involved in bringing all these individual building blocks of the monoprotein molecule together in a very defined and linear process, starting from this core molecule called myristicin, stepwise addition of sugar molecules, the addition of this caffeic acid side chain, a few more sugar additions, in this 6-step reaction coming from this simple building block here to this very specific complex molecule. And this work is published, and it's not the topic of a talk like today. What we really could use for the discovery strategy, and I think this was a key recipe to be successful in this, exploring the biology of this plant, specifically the spatial and temporal pattern of plant development, how plant development is associated with production of certain molecules. The spatial pattern is one where across all the different parts of the plant, it is only these below ground storage organs that are bulb-like, called corms, that accumulate this molecule. This was the spatial feature that we explored. This is where the molecule is present. And we explored a temporal pattern as we followed the development of these corms over the course of a growing season from spring to fall. The old corms that we would plant now are over the time of a year, and then developing young corms. So it's a bit like a potato would grow. Here's your old corm or the old bulb. It develops these below ground shoots, and at the end of which is where the above parts of the plant grow out. But also below ground, it starts to develop these young corms at the base of each of these plants. So each of these old corms or mother corms gives rise to about half a dozen to a dozen offspring corms. And they grow over the growing season. We call them young corms. And it is only in the young corms where we could actually detect a temporal pattern of increased accumulation of this molecule. That was also then confirmed by enzyme activities. This is the time and the place in the corms, and only during the early summer where this biosynthetic pathway is active. And we utilize this information about the spatial and temporal occurrence of the molecule to filter through these very, very large gene sets that we got from transcriptome sequencing, filter specifically for candidate genes that match this temporal and spatial patterns. And in this way, we discovered actually the enzymes that are essential and sufficient for the accumulation of individual building blocks into this very, very specific molecule. So this was as much technical detail as I talk about today. And again, most of this work is published and the references are available in this presentation. Now we come to strategies for production. And here's where I have some results, but we also want to discuss a few things, and builds in part on what Vince Martin talked about last week. We're now thinking about how to produce this compound in a scalable fashion, such that we can actually go into the development of a nutraceutical or pharmaceutical, assuming that the clinical trials will be successful. Well, the first and most obvious one, plant this Montbrechia on large acreage and develop all the tools around it to develop it into a crop. It's only being grown now on very, very small acreage in the Netherlands as a horticultural production system, just for bulk production for garden centers. Another option could be, and this is what I think we had a discussion about last week, exploiting plant cell cultures. Maybe we can actually derive plant cell cultures from these corms. And if they don't produce naturally, maybe they can be elicited to produce this molecule. And this would be following what is still being done in part for Taxol production as this famous anti-cancer drug. So those would be plant-based production system working with the endogenous system or native system. But here would be heterologous or bioengineering systems where we would either use yeast as a production platform, using the type of technologies that Vince Martin explained in detail last week. Alternatively, use plant metabolic engineering. And here we would go into Nicotiana benthamiana. It's a plant that is used in the industry for production of therapeutic proteins already, and would have this capacity as a scalable system. I will just give you a few snapshots at what we've been doing in these two systems, Nicotiana benthamiana and yeast so far. And again, this is a highly simplified summary of research to date in Nicotiana benthamiana, for now using transient expression by agrobacterium-based vacuum infiltration. We've been able to transform this plant with the 6 essential assembly genes for this molecule, plus a few additional genes that are essential for producing this core part of the molecule. And we are able to transiently produce MBA, monoprotein A, in this Nicotiana benthamiana system. So this is proof of principle, at least, that a plant that would naturally not produce this molecule can be transformed in its leaves, which is a rapidly growing tissue, to produce this very rare metabolite. But there are challenges still. One of our observations was that in addition to this molecule, monoprotein A, that's our target molecule, these plants also produced a molecule called MBB. And the only difference between monoprotein A and monoprotein B is one oxygen atom. And it's one oxygen atom that is different in a caffeic acid side chain or in a coumaric acid side chain. And this one oxygen atom difference in this molecule renders MBB entirely inefficient as an inhibitor for the pancreatic alpha amylase. It has a Ki value that is almost 4,000 nanomolar. So this is not by any means anywhere close to an efficient inhibitor. And the reason why the Nicotiana plant produces those compounds was because it draws from metabolite precursors that it has in its general biochemistry. And we realize now that we do not only need to concern ourselves with how this molecule is being assembled from its building block, but we also need to engineer the precursor biosynthetic pathway steps. And all these precursor biosyntheses are established in Nicotiana. That's why it's a good chassis system for us to work in, but we need to tweak towards which precursors we make available, kaffeoil and not coumaroil. Now this leads over to yeast. If we go into yeast for bioengineering or metabolic engineering or bioengineering of this molecule, we can in the same fashion transform yeast with these 6 genes and enzymes for the assembly of this molecule from its building blocks. But in yeast, most of these building blocks do not naturally occur. So in addition to engineering yeast for the building block assembly pathway, we also need to engineer into yeast the capacity to produce phenylpropanoids that yield precursors such as caffeic acid. We need to engineer the flavonoid pathway, which produces this myristicin, and some of the UDP sugars that are essential for the biosynthesis, which makes yeast a much more complicated system to engineer, or it makes engineering in yeast more complicated. And we are approaching this, we're using a modular cloning approach here. In total, we need to engineer somewhere between 15 and 20 genes into yeast, and we divided those genes into modules of smaller sets. One goal is to produce the flavonoids, including specifically myristicin. Another set of genes is needed to produce the UDP sugars, specifically producing UDP rhamnose and UDP xylose that we do not have in sufficient amounts in yeast. and then another module to engineer, another set of modules to take all these building blocks and assemble them into MBA. And the beauty of a modular approach is that we can actually test many different combinations of genes, gene variants, as well as promoters. And that again leads back to what we heard last time in Vince Martin's talk already, where if we had the capacities of a foundry, this would give us a lot more capacity actually to test all these possible combinations. For the development phase, we still need to feed precursors and intermediates, but eventually the goal is to bring all the modules together such that we can produce all the precursors, all the intermediates, and the final product in yeast. And here's just one example, final slide here today in what we've accomplished so far. This is just showing an example of two modules that were co-expressed in yeast, each containing 5 genes. We still need to feed myrcene caffeic acid, but we can also produce these molecules with different, these building blocks with different gene modules. We actually now seeing in yeast the production of 1-protein A. So proof of concept, proof of principle is established. And that's where I think we're ready to plug in with bioengineering. Here's just acknowledging all the other students, postdocs, research assistants in our group who've been involved with the work so far. I want to acknowledge Plantform. Plantform is the Canadian-based company who we are working with using their Ecosiana Bentamiana platform. I want to acknowledge Dr. Max Planck at Stellenbosch University in South Africa. We are working together with Dr. Makunga on developing the cell culture production system. And our funders, which include GlycoNet, a network of centers of excellences based at the University of Alberta in Edmonton, and Canada's NSERC. Thank you.
Joerg, thank you so much. I gotta, since I moderate, I get to ask the first question. It was interesting on the temporal expression of the metabolite in the new corms. What do you think the purpose of it is? Does it inhibit the plant's own starch degradation as it's assembling the corms, or does the plant have amylases that are resistant to the inhibitor? I wonder. Is it a defensive mechanism?
Mm-hmm. Yeah, we do not know. That's actually a really good question, Steve. We do not know how this molecule affects carbohydrate metabolism in the plant. Our hypothesis is, and this remains to be tested, is that this molecule is a defensive compound and functions perhaps in a context of plants interacting with herbivores, thinking of this organ as a vegetative storage organ, but also a part of the plant through which it actually propagates year over year, and the organ to bridge the non-growing part of the season. I would say overwintering, but in its native environment, it would not necessarily be overwintering. The plant accumulates starch in these tissues and stores it there for some time while not actively growing. And this is a tissue that could potentially be a nutrient resource for small rodents, for example. Right. And well, we know from our work in the animal test that it very efficiently affects starch degradation in rats. So presumably it would also do the same in other small mammals that may actually feed on these molecules. And it's been known shown in research in this field of chemical ecology that animals have a learning capacity. If they feed on a plant, as nicely as it may taste or whatever, if they don't receive nutrients from it and energy, they eventually will stop feeding on it. They go somewhere else. So that's our hypothesis, that it is actually an undefeated compound.
Okay, which makes that interplay between plant and herbivore, whether it's an insect or an animal, that does make that's an interesting pathway hypothesis. One of another question is, you mentioned the modular approach to expression of the compound. Are any of the genes expressed in other parts of the plant?
Yes. So generally, just try to pick this out. So general phenylpropanoid biosynthesis. Okay, so let me just take a step back.
Yeah.
The genes that are involved in the assembly, most of them are very tissue specific to these developing columns, both with their spatial and temporal patterns.
Okay.
This is, of course, I mean, it's a bit of a circular argument, at least initially it was, because that is what we used as a discovery strategy. We used selective spatial and temporal pattern to discover these genes, as these patterns match the accumulation of the molecule. But then we went back with gene probes after cloning these genes. We went back to gene expression analysis across the plant. We found that there was no substantial expression of these assembly genes in other parts of the plant. Now, the building block biosynthesis, those pathways are actually more ubiquitous across the plant, and also quite a bit really ubiquitous across the plant kingdom.
Yeah, like the phenylpropanoid pathway, one of my favorite pathways for like vanilla, and also for glucosinolates, if we are talking about Canada's canola, sinapine and other kinds of interesting metabolites. Another question is, what do you see with I like the approach of the precursors in the assembly. What do you see as the limitation to heterologous expression, or expression to get to the levels that would be needed as a therapy, or as a treatment, I guess, not a therapy, it's a treatment for type 2 diabetes? What do you see as kind of the next steps, Joerg?
So the limitations and next steps are different in the two systems that we are actively pursuing now, which is Nicotiana benthamiana and yeast. In Nicotiana benthamiana, it is primarily redirecting precursor biosynthesis such that we make predominantly caffeoyl-CoA available for the availability of this side chain here, caffeic acid, as opposed to getting coumaric acid or coumaryl-CoA, which would lead to an ineffective. So there's a real competition in this pathway. We need to get away from coumaryl to get into the side chain here, but at the same time, coumaryl is itself a precursor for this myrcene core.
Okay.
So this is actually our biggest challenge right now, directing precursor flow in Nicotiana benthamiana. And that's why we need to go back into Nicotiana benthamiana's endogenous precursor biosynthesis and manipulate that, plus put some additional genes from one pressure in here to affect myrcene biosynthesis. So that's the biggest challenge there. For yeast, it is the number of genes that we need to engineer, somewhere between 15 and 20 genes from plants that need to be moved into yeast, including the precursor biosynthesis and the assembly genes. The assembly genes work well in yeast. Precursor biosynthesis is more of a challenge. And then in yeast, and I believe Vince also referred to this last week already, compartmentation. Plant cells have multiple compartments, some of which are plant specific, like plastids, where some of this biosynthesis occurs, and have plant tissues have specialized cell types or tissue types that can be utilized for specialized metabolism to be sequestered away from housekeeping functions, accumulation of metabolites that may potentially interfere with endogenous metabolism. This could potentially be an issue in yeast.
Okay. Another question that has come up, and again, for folks in the webinar, use the Q&A session or section to type in your response. It relates to kind of something that Vincent and you just touched on, Joerg, about the is yeast the right chassis to express this in? And I know you have two different strategies, which I think plays to some of the, and each has challenges. So what do you see as yeast? Is yeast a good model, or is it a production system?
Well, we're not close to a production system yet.
Right.
I think it has more potential than just a model for discovery, and I would not want to decide between those two possibilities now: the plant metabolic engineering and plant bioengineering, and the yeast bioengineering, because I think that both systems have a lot to offer. I'm not trying to like give a diplomatic answer here.
No, I know.
And not commit to anything. If we wanted to develop a company around this, we would probably have to, and we want to develop something around, we will have to commit to one or the other. But I think they have very different opportunities, these two systems, and I really want to explore them most. And again, listening to presentations last week and the first presentation in this seminar series, bioengineering has so much to offer in new tools and techniques for engineering microbial hosts that I think we want to be in line with this molecule there.
Yeah, I think that I agree, and I think at this stage you want to keep all your options open to explore the possibilities, and we don't know what we don't know, and it's a way to gain insights into how to produce. And I'm thinking back to, like you mentioned, back to Patrick's Westfall's presentation at the beginning and Vincent's. I think one of the opportunities is, again, to be thinking about an opportunity for engineering biology is to onboard other systems onto this type of platform, again, to take advantage of some of the precursors that are made in the host. So again, I think those are really those are good things for us to consider. And again, back to the biodiversity, the chemical biodiversity that you introduced today, I think is, if in Canada, if we had the goal of generating many more potential metabolic metabolites and insights into metabolic pathways as a national resource for use in bioengineering, how should we target the huge range of plant resources for sequencing and further analysis? Is there would you have a thought on how would you prioritize and schedule that?
Yeah, that's a great question. One would be, of course, like metabolites that are already known, that have known functions, that have known biochemical pathways, where the genes are known, where what's left is a difficult part of reassembling these pathways and reconstructing them in a heterologous system that can really be bioengineered to production scale. And those would be priorities, obviously. Yes, we know what Taxol looks like, what the activity is for Taxol, how Taxol is being biosynthesized for most of the steps, actually. But it's still, I think, the holy grail for, can we reconstruct Taxol biosynthesis in a bioengineered system? So this would be one out of those many examples where pathways are known, activities are known, where we actually need to work on the bioengineering side. I would go back then to examples like this one, where an activity is known, we have the opportunity to discover biosynthetic pathway, and then take this into the same type of production pipelines. But I would also go a step back, so this is in terms of prioritizing, exploring traditional knowledge of medicinal activities or other useful activities that we consider useful, explore traditional knowledge. This can be traditional pharmacognosy, so the knowledge of medicinal plants. This can be literature that's available that is joining in research and development with indigenous people's knowledge. For example, And this has to be done very, very carefully and cautiously, and in a mutually benefiting way. And then broad screens. This is what led to the discovery of the biological activity of this molecule 1.8. The molecule was known, it was described in the natural products literature, but its biological activity was not known. So screen the existing chemical diversity or biodiversity. Vince Martin and I and several others have engaged in projects like this that dealt at least with part of these prioritizing activities. Phytomedicine was a Genome Canada funded project, which I think was enabling some of those capacities that Vince has since developed and that we and others developed. Actually, I should say, interestingly, Steve, Saskatoon has a very, very strong and successful history in this. Most recently, work by Pat Cavallo that contributed to the knowledge of the biosynthesis of artemisinin, which was then be utilized at Amaranth for artemisinin production, but also going back in the 80s. The Plant Biotechnology Institute at the NSC in Saskatoon was one of the leading places in the world for exploring plant cell cultures for specialized metabolites. So much of the work, and I think Vince rated this too last week, on the anti-cancer drugs vincristine and vinblastine from Catharanthus roseus. That work was to some large part done in Saskatoon at the Plant Biotechnology Institute in the 80s.
Yeah, it's amazing how one of the things that's exciting about the engineering biology platform, and I like how you mentioned the knowledge, the traditional knowledge, working respectfully and appropriately with Indigenous communities to mine, but even the work that was done in the, the cell culture work that was done in the 80s, it's like there's new tools, new approaches to go back and to be able to bring forward products, and that skill set, that plant tissue culture skill set is important today, or re-important today, as it was back in the 80s. And I think it's one of those things that how do we maintain that capacity, capability, and expertise? Because it's a competitive advantage for the institutions and countries to be able to offer new solutions. One of the things that I also liked about your seminar, and I'm going to say this because my good friend and colleague Kirk Brewster's on the call. He's a medicinal chemist, a synthetic organic chemist, and it's that partnership, Joerg, that is necessary to be able to elucidate what is going on here, to be able to create new opportunities. So I really like your presentation and the absolutely essential requirement for that partnership. And I'm glad you guys talked in the hallway. And I'm sure Steve was glad it was just the spiral diagram that was all that was needed to kick off this really excellent research that you've presented here today. So again, I think that multidisciplinary aspect is another message I think that's really important for everybody on our webinar to hear. If there's no other questions from the audience, I do want to thank you, Joerg, for presenting. And just kind of in closing today, with this webinar today, this brings to an end our exciting series, this season's exciting series, because we do have some other opportunities that we're going to line up. All of the videos, and all the recorded presentations on this series are available on GIF's YouTube channel. This one, Joerg's, is not there yet. We'll have to upload it, but we'll get it there. And again, we'll continue to share them via our social media channels, so you can watch and rewatch and catch up if you've missed any of the previous ones. And Joerg did a great job of mentioning, and really, he was the perfect speaker because he tied in previous talks into the application and the opportunity around engineering biology. Joerg, on behalf of the Global Institute for Food Security, thank you so much. And I want to thank our organizing committee, our Director of Communications, Funke, David Conlon, in our communications group, and Tina Murdoch for helping organize and deliver this webinar series and making sure that I showed up on time for all of these wonderful, wonderful presentations. So thank you, everybody. And with that, I wish you a safe and happy day. Take care.
Great. Thank you.
And Joerg, I've got an idea I want to talk to you about.
Okay.